Cancer treatment by polymeric micelles. Hello, I am Adnan Murad Bhayo, and in this presentation I will focus on one of the main issue of the world, that's the cancer. Cancer figure among the leading cause of mortality worldwide with approximately 14 million new cases and 8.2 million cancer related deaths in 2012. This was a report which was released in 2014 by the World Health Organization, and we know that the cancer is the second largest disease of the world after the cardiovascular diseases. And in this report you just compare the ratio between the mortality rate and the new cases. There are 14 million new cases and 8.2 million cancer deaths. So this is roughly the 60% deaths in the 2012 due to the cancer. So what is the reason behind this? As we know that we have hundreds or even thousands of the drugs of the cancer are present, but cancer is not in control. So why? Because the simple reason is that your drugs are hydrophobic in nature. Let's take an example of the oil in water. You just put one drop of oil in the water, it cannot be dissolved because your oil has hydrophobic properties and water is a hydrophilic property, so it cannot be dissolved. Hydrophilic will dissolve in hydrophilic and hydrophobic will dissolve in hydrophobic part, like dissolve like phenomena. So your drug is hydrophobic and your blood is hydrophilic in nature. When you inject your drug into the bloodstream, it cannot be dissolved because your blood is hydrophilic in nature. So you should have some carriers for these type of drugs. So problem is the solubility of these drugs and the solution from the polymeric micelles. So what is the polymeric micelles? Polymeric micelles is a macromolecular assembly that forms by the self-assembly of blockopolymers that has the inner core and outer shell. So simple we can say that the blockopolymers has dual properties. That's it should have the hydrophobic parts, which we say that is the corona, and the hydrophilic hydrophobic core and the hydrophilic corona. So what hydrophobic do? Hydrophobic simple that you inject the hydrophobic drugs into this part, and the hydrophilic part will enables the drugs to move into the bloodstream. Like you take an example of the bot acts as the micelles. Your water, here is a water is a blood, and this is the hydrophobic drugs. So the micelles should have dual properties. So amphiphilic blockopolymers, it has dual properties, that is a hydrophobic segment and the hydrophilic segment. When it dissolves in the water, it forms these type of micelles. What the chemistry behind these micelles I will discuss in another slide. So you just take an example of the blockopolymers, like this is polyethylene glycol, it has hydrophilic segment, and another is polycaprolactone, which is hydrophobic. You just observe that CH2 higher number of CH2 group makes it hydrophobic. So this polymer blockopolymers I will discuss on this, which has dual properties, that's hydrophilic and hydrophobic. When it dissolves in the water, it forms the micelles, this type of micelles where we inject your drug of interest and enables for the treatment of the patient. So what is the chemistry behind the micellization? How the micelle is formed? You should know about the surface tension of the water. Why surface of the water is curved in shape? Because you observe that in the inner layer, water molecules has the force from all direction, like from here, like from here, like from here, like from here. So every direction should be cancel each other. So this layer should be in this type, no curve shape. Where you observe in the surface of the water, it has the force from this side, from this side, from this side, from this side, but not from this side. So this force, the molecules from here attract inner side, so it makes it the curved like shape. Now what the blockopolymers, which has two types of properties, do in this form the micellization. So when you put the one drop of these type of blockopolymers in the water, so blockopolymers has hydrophilic and hydrophobic. Hydrophilic head will interact with the surface like this, and hydrophobic will should be away from the surface, because nature of the water is hydrophilic, and nature of another, there's the polycaprolectron which is hydrophobic, should away from there. And the drop, and it makes the surface like this, like this, like this. When this surface is complete, you add another drop of the drop of these block, it should go towards inner side. And what the phenomena will do here, so you observe that this is a hydrophilic part will interact with the hydrophilic water, and the hydrophobic part should move away from the water. So this type of structure are formed, like you observe that if this type of micelles are formed. This is a micelles. So what are the properties of this micelle? I took just one paper for example. Micelles of methoxy polyethylene oxide B polycaprolectron as a vehicles for the solubilization of controlled delivery of cyclosporin A. So what the properties of these micelles? Micelles should have very small in size. Simple, you inject your micelles in your body, and it carries your drugs of interest in every part, like you inject the drugs for the kidney, so it should be passed from the kidney. So nephrons has very small in size, I think 0.2 to 10 millimeter in size. So your micelle should have very small in size, as it pass from the kidney and also from the renal excretion after they perform their function. So should have very small in size. In this paper I just observed that the micelles have the size 87 to 99 nanometer. So 10 to 100 nanometer is very satisfactory for the drug delivery. And the important thing is large amount of drug loading. So drugs load on these micelles are very large, like the 48 to 63 milligrams per liter. You can load the drugs in this micelles. You cannot load the drugs in another carriers in this amount. 48 to 63, very high amount of drugs, this one. And the important is that the polymeric micelles have a very low toxicity, because you inject another, we compare the polymeric micelles with another carriers, chemophore EL. When chemophore EL used as a drug delivery carriers, it has two type of limitations, safety limitation. There's a hypersensitivity in which the immune system develops, like you observe that the allergy patients. And another is the nephrotoxicity, that's simple, that's a kidney failure, because the size of these carriers is very high as compared to the nephrons. So nephrotoxicity is also observed here. So but these micelles, I just read all the research paper, and these micelles have no or very simple low toxicity. So another thing is drug release rate. So it's a very important. If you just treat the cancer patient, this treatment is not for a 1 day, for a 2 day, it's a long-term delivery. So you should have the carriers which has drug release very low drug release rate. Like you observe that I just observed, sorry, in this research paper, that's after 12 hours chemophore EL, that's a carrier, 77% of the drugs is released by these carriers. So but the polymeric micelles has a 5.8% after the 12 hours, so it can be used for the drug release rate. So at the end I just say that the polymeric micelles are very important for the treatment of many diseases, and most probably the cancer, as compared to and the carriers. Thank you very much, and for more videos you can subscribe my channels.